Alright, so the very last part of section 6.3 is about the Golgi apparatus and the Golgi apparatus and the rest of the endomembrane system. So remember that the endomembrane system is everything inside the cell that's composed of a phospholipid bilayer membrane system. So it's the same material that makes up the cell membrane, but instead it makes up a lot of different kinds of structures that do different jobs inside the cell. So the ER, both the rough and the smooth, is part of synthesizing materials and sending them to where they need to be, as well as modifying them. So the next step is the Golgi apparatus. And the Golgi apparatus looks very much like the ER in pictures because it is a bunch of flattened sacs made of a double membrane, just like the ER is. So after things are, these sacs are called cisternae. And cisternae stands for, means something. A reservoir for liquid. That's what cisternae means. So all these sacs are called cisternae, and we can call the sacs of the ER the same thing. So if you hear that, that's what that means. And after leaving the ER, the transport vesicles that we talked about that just came off of the transitional ER, they go to the Golgi apparatus. And if we are going to talk about what the job of the Golgi is in terms of the whole scheme of the cell, it's kind of like the post office of the cell. Get things, like finishes things up and packages them and gets them ready to be shipped off somewhere else. So since it leaves or makes things to send to other parts of the cell, there's a lot of Golgi apparatus inside of secretory cells, just like we said there was with ER, because the ER is where these things that are going to be secreted are made, and then the Golgi is where they are prepared to be sent away to other parts of the body. So it's important to know that the Golgi is two-sided, and we talk about the two sides differently. So there's the side that accepts transport vesicles and the side that makes and ships off transport vesicles. So we say that the Golgi has a polarity, much like a water molecule. The two sides are different, and that's what that means. So this right here is the cis face of the Golgi apparatus, and that means it's the receiving end. It's where the transport vesicles from the ER arrive at the Golgi, and then over here is the trans face, where things get shipped off from the Golgi apparatus. So that's what you see here also. You can see all the flattened sacs. I believe this is the cis face. Right here's the trans face where they're sending it away. So the cis face is the receiving end, and it's located near the ER. So all of these little transport guys are coming from the ER, and they arrive and they blend in with the cisternae, or the membranes of the Golgi apparatus. So you can see how as something arrives, it just kind of opens up, and these little purple guys just become part of the Golgi apparatus. And then once everything is ready to go, it's shipped off and packaged in these little transport vesicles coming from the trans face of the Golgi apparatus. And so basically, the Golgi receives things from the ER, fixes them, switches them up a little bit, and then sends them away. So one thing that the Golgi might do is it might modify the carbohydrates that are on the glycoproteins. So remember that glycoproteins are proteins that have a carbohydrate attached to them, and the carbohydrates were added inside of the endoplasmic reticulum, and then the Golgi is going to take that carbohydrate, it might add some sugar monomers, take off some sugar monomers, do whatever it needs to do to get the product perfect and ready to go, and then it's ready and it ships it off through the trans face. The Golgi might also make some macromolecules. Because many polysaccharides that are secreted by cells are Golgi products. So polysaccharides, remember, are the polymers of carbohydrates, and so these guys are made inside the Golgi and are shipped off, again, by the trans face. And so the products that are ready to go, like I said, they're going to leave via the trans face of the Golgi apparatus, and they will go and they'll find the plasma membrane and they'll fuse to it and open up outside of the cell. So that way whatever is made has now left the cell. Just like here, these guys came and fused with the Golgi membrane. The transport vesicles that leave from the transphase are going to go and fuse to the plasma membrane and be outside of the cell. And so before the transport vesicle leaves the transphase, the Golgi is making sure that they are labeled correctly. So they might add like a phosphate group, or they might add some other sort of group that's going to act as a signal to send the vesicle to where it needs to be. So these signals, these messengers kind of act like zip codes. If it were going to be a post office, so it's sending it, you put a zip code on it so it knows which part of the country to send the letter to. Whereas here, it puts some sort of signal on it so it knows which part of the cell to go to. If it needs to leave the cell, it's going to go to the ER, if it's going to go to a lysosome, whatever. So we put a phosphate group, some sort of signal, onto the vesicle to send it to the right place. Then, the next part of the endomembrane system is what's called a lysosome. Lysosome is it's like the digestive compartment. So this is where breakdown of things happen. So by definition, it's a membranous sac of hydrolytic enzymes that can digest macromolecules. So again, made of the same membrane as the ER, of the nuclear membrane of the Golgi apparatus, made of exactly the same things, except it's not quite a series of folds. It's just like one bubble, and it has hydrolytic enzymes. So it's important to know that these enzymes are hydrolytic, meaning they do hydrolysis, meaning they break the bonds found in the macromolecules. Just like hydrolysis, we put in a water molecule and we break the bonds to break them down into their parts. So if we have a macromolecule that we don't need anymore, and you put it into a lysosome, combine it with the hydrolytic enzymes, hydrolysis is going to happen and it's going to be broken down into its monomers. So that's the job of the lysosome, basically. So these, the hydrolytic enzymes work best at a very low pH. So the pH is different inside of the lysosomes than inside of the cytosol, and so that's important. The enzymes only really work inside of the lysosome, so that's why they're contained. If they're released, then they don't really do their job. They're kind of neutralized by the pH of the cytosol. But if you were to release a bunch of lysosomes at the same time, like release their contents into the cytoplasm, the cell would digest itself and die, which sounds scary, but sometimes is on purpose, and we'll get to why that might happen later. But anyways, these enzymes inside the lysosome are used to break down the macromolecules that the body does not need anymore. And then it can use the parts to do something else. So let's say that we didn't need this one protein anymore. You might send it to the lysosome to be broken down into the amino acids, and then the amino acids would go elsewhere and build new proteins. So we're not destroying things, we're just breaking it down to be recycled into something else. And the hydrolytic enzymes that do this, as well as the membrane of the lysosome, are made in the rough ER, and then they're sent to the Golgi apparatus for processing. So even the parts of the endomembrane system are made by the endomembrane system. So it's just very continuous. So lysosomes do digestion, like I was saying. So one of the processes that they do is certain single-celled organisms like amoebas or other protists, which we'll talk about later, but other single-celled organisms, they eat by actually like taking in a particle of food. This is called phagocytosis. And so this is when a cell takes in another cell, and then what we have is what's called a food vacuole. So vacuole is a storage place, and so if you're like grabbing a piece of food and pulling it into your membrane system, then you have a little storage section inside of you that has food in it. And that food isn't going to do you very well unless you are able to break it down. So what happens is, do I have a picture here? Okay, so here the cell is grabbing a piece of food, bringing it inside, and then now it has this food, but what good is that food unless we can do something with it, right? So this food vacuole right here would fuse with a lysosome. So anything that's part of a membrane system, they can just fuse with each other if they want to. And then now we have all these digestive enzymes that can perform hydrolysis. So let's say this is a starch molecule. This is a starch molecule. We want to break it down into the monosaccharides that are found in the starch molecule. So we need these hydrolytic enzymes to perform hydrolysis on the starch molecule, and then we can have monomers that we can use for something else. So that's one thing that they do. Some human cells do phagocytosis also, like our white blood cells. If you've ever seen like a video of a white blood cell kind of grabbing an invader, a bacteria, and taking it in, that's phagocytosis as well. And then we also will use the hydrolytic enzymes, this is what I've been talking about, to recycle old macromolecules that we don't need anymore, or old organelles that we don't need anymore. And it's called autophagy. That's how you say it, right there, autophagy. So we're eating ourselves, basically, because we don't need it. So it might be damaged, or we just have an excess of something, but we would make sure that it finds a lysosome and get broken down into its constituent parts. That way we can use those again, and it's not a waste. So here is a vesicle, so with a mitochondria and a peroxisome inside. So we'll talk about what both those are, but this cell doesn't need those anymore. So it wraps them in a membrane, and now that membrane can easily fuse with a lysosome and break everything down into parts that can be used again. And then lastly, we have what are called vacuoles. Vacuoles are basically storage closets inside of the cell. So, or diverse maintenance compartments. So there's several different kinds of them. A plant or fungal cell may have one or several vacuoles derived from endoplasmic reticulum and Golgi apparatus. So it's a membrane-bound organelle with one of many things inside of it. So the first one, it says, are food vacuoles, which are formed by phagocytosis. So these are basically those little bubbles that can store food inside of them. And the likelihood is that they'll probably form with a lysosome, so we can break down the food and use it for something else. Then we also have contractile vacuoles, which are found in a lot of freshwater protists. These are just freshwater, single-celled organisms, and they use it to maintain the water balance. So they live in freshwater, so they have to maintain the correct solute concentration from themselves and the water, because they can't be flooded with too much water. They need to keep all the right nutrients inside of them, so they use contractile vacuoles to pump water out of their cells. And then plants have what's called a central vacuole, which you guys are probably familiar with. This is just a very large membrane which they store water in, and it helps keep pressure against the cell walls and helps keep them standing upright, keeps their cells what we call turgid. Otherwise, if a flower starts to wilt, or a plant starts to wilt, it's because it's running out of water, so it's starting to take water out of its central vacuole to use for metabolic processes, such as hydrolysis, because it needs water. So many plant cells are made up of mainly a central vacuole, and that's what that is right there. It can also store pigments. So in plants, if you see like red or green, or red or blue or pink flowers, it's probably because they have those pigments stored inside of the central vacuoles in those plant parts. It can also store organic components, so like monomers they might need later. And plants that are poisonous often store their poison inside of their central vacuoles. That way it won't get to the plant parts that need it. And so basically, to end the endomembrane system, we have the nuclear membrane, the plasma membrane, so the cell membrane, endoplasmic reticulum, both rough and smooth, the Golgi apparatus, lysosomes, and central vacuole. And it is a complex and dynamic player in the cell's compartmental organization. That's what you need to know.